It is a pleasure for me to introduce you to Michael Thon. Michael is from Palatine, a village close to Chicago, and he studied his degree and master in botany at Eastern Illinois University. He then did his PhD in plant pathology at the Pennsylvania State University, and then he went for a first postdoc at the University of Kentucky, where he started to work with the mice anthracnose pathosystem, studying several virulence-related genes of Colletotrichum graminicola. He then went for a second postdoc at North Carolina State University, where he developed his interest in fungal bioinformatics and genomics, working on the genome sequence of the rice blast pathogen Magnaporthioriaceae. After 3 years period at Texas A&M University, he obtained in 2007 a Ramon y Cajal Fellowship, and he moved to Salamanca. And he's now there, full professor, catedrático, and at Universidad de Salamanca and CIALE, Instituto Hispano-Lusode de Investigaciones Agrarias, in the Department of Microbiology and Genetics. And he leads there the research group on fungal genetics and bioinformatics together with Serenella Sukno. And during this time, they have been doing very relevant work on genomics and genetics of fungi, especially of Colletotrichum. So I leave you with his very interesting talk about genome evolution of Colletotrichum plant pathogens. When you want, Mike.
Okay, thank you very much, Salida. First for the introduction and also for the invitation to give a talk to all of you. I wish I could visit in person, but we all know that cannot work right now. But hopefully someday I can go over there and visit you. So I'm going to talk about some of our work on studying the evolution of genome, or the genome evolution of anthracnose-causing fungi in the genus Colletotrichum. First, I just wanted to show you on my cover slide here a photograph of our institute. This is the institute for the Instituto Hispano-Lusode de Investigaciones, Agrarias. Our building is, I think, about 10 years old, and it's situated here in a research park on the banks of the Rio Tormes. In fact, my office is right over here, and if I turn around, I can look outside and I have a very nice view of the river, which is very nice. And as I said, over on this side of the building, we have mostly offices, and then all the research facilities are over here on this side. This building houses 7 research groups that study a broad range of subjects related to agriculture. Molecular genetics, of plant microbe interactions, of beneficial fungi, plant physiology, water resource management, etc. So let's continue. And with the next slide. And as I said, we work mostly on the genus Colletotrichum graminicola, which is the maize anthracnose. And you can see a photograph of the lesions on a leaf here at the bottom. But Colletotrichum is a very large genus, and it causes diseases on a wide variety of different plants. Almost every agriculturally important crop has a pathogen from the genus Colletotrichum that infects it. And as you can see from this slide, a lot of the diseases are post-harvest diseases. And many of them are very economically important. So as I said, we focus mostly on Colletotrichum graminicola, which causes maize anthracnose. You can see some of the symptoms here. The stalk rot, you can see the lesions on the stems here. You can see the lesions on leaves. It also causes a disease called top dieback. In fact, the fungus can infect all parts of the plant, but these are the diseases or phases of the infection that are economically the most important. So we use Colletotrichum graminicula as a model system for studying the evolution of pathogenicity in plant pathogenic fungi, but it's also an economically important disease. And this photo, it was taken in 1973, I believe, in Iowa. And you can see that this crop of maize is completely destroyed. And this is due to the stalk rot phase of the disease. The fungus infects the stalk becomes very weak, and the plants basically fall over. And even though the ears, the mazorcas, are perfectly healthy, they cannot be harvested in this way. The harvester cannot collect them. So it can cause big economic losses. And in fact, there are areas today in the United States that cannot grow maize because of this disease. The farmers simply cannot grow maize because they'll lose their entire crop. So in 2012, we published a paper describing the Colletotrichum graminicula genome. This was a consortium that started it back in 2006. It was funded by the National Science Foundation and by the United States Department of Agriculture. And in this slide, we just have a summary of some of the statistics. The genome is about 50 megabases in length. And we have 653 scaffolds. And in fact, this was sequenced mostly with Sanger sequencing and Illumina sequencing. And what we have recently done is to resequence this genome using PacBio sequencing technology. If you don't know what PacBio is, it's a sequencing technology that produces very long reads, okay, up to tens of kilobases in length, and that really helps to make long scaffolds. And in fact, now I think we have 20 scaffolds instead of 653. And several of those actually are telomere to telomere sequences. So we have complete chromosome sequences, at least for some of the chromosomes. So this is just a little index of what I'm going to talk about. And I'm going to talk about three different research projects that we have been working on and continue to work on. First is gene content differences in Colletotrichum species. The second is intraspecific genome rearrangements in Colletotrichum graminicula. And finally, a relatively new project for us is to study population genomics of graminicula. And we're learning some very exciting things about the population structure of Colletotrichum graminicula, which I'll show you. Okay, so this is just a phylogeny of the genus Colletotrichum. As I said, it's a large genus. It contains actually more than 120 species. Most of the species, as you can see, belong to species complexes, and that's what these boxes represent. A species complex doesn't really represent any formal taxonomic rank. It's just a convenient way to refer to groups of phylogenetically related individuals. What I'm going to focus on for the next few slides are two species complexes, the Gliosporides species complex and the Acutatum species complex. And the one thing I want to point out is that they're practically on different sides of the phylogeny here, so they're not very closely related, and that'll be important in a minute. So one of our interests is studying the evolution of host range of pathogenic fungi, and we can classify pathogenic fungi into, or we can try to classify them into three groups: the specialists that have a narrow host range that infect only one species or maybe only one genus. There are the generalists who have a broad host range who can infect a broad range of different genera and different species. And then there are these transitional species that infect, that have an intermediate host range, and they only infect a few different species or a few different genera, or maybe members of one family, etc. So the important thing here is that the Acutatum and Gliosporides species complex in general are generalists. Most of the species in these two groups are generalists. They have broad host range, but not all, as we'll see in a minute. So for the next few slides, I'm going to show some research that we did with the species that you see in this table. These are all strains of Colletotrichum and also some non-Colletotrichum species down here at the bottom that we included as sort of outgroup species. All of these at the time that we started this had genome sequences available. So one of the first things we did, and I should point out that all the next slides are the work of Riccardo Baroncelli, who is a former postdoc in our group and who we continue to collaborate with. And a lot of this work is, well, some of it is published and some of it is in a paper that we are working on right now. And so this is one of the first figures that Riccardo made in studying these different species. And you can see that there are differences in the number of genes encoded in the genomes of the different species. But what was interesting here is that if you represent the secreted proteins as a percentage of the genome, the species that have the largest secretomes are the hemibiotrophs. So biotrophs are these fungi that can infect the host without killing the host cells, essentially. Necrotrophs, on the other hand, kill host cells, and they cause the big necrotic lesions that you see on leaves and other part plants. Hemibiotroph is basically a fungus that begins life as a biotroph and then switches over to a necrotrophic lifestyle. So we can speculate that maybe the hemibiotrophs have more secreted proteins or a larger diversity of its secretome because it has more diverse needs. It needs to live two different lifestyles. So another sort of exploratory analysis that we did is to classify the proteins into families based on interpro. And in this heat map that we see, each row represents a different functional category. And each column is a different species. And then each box in here this is a heat map, and the orange and red colors represents overrepresentation of a certain family in a certain species. And what's interesting here is that this box here at the bottom highlights the members of the Gliosporoides and Acutatum species complexes. So we see a big overrepresentation of certain gene families. Well, what are these families? Most of them have roles in carbohydrate metabolism and protein degradation. So it seems like these are families that are expanded in both the Gliosporoides and Acutatum species complexes. So we did more detailed analyses of this phenomenon, this expansion of these families. And this time we classified the proteins using the CAZyme database. The CAZyme database, or CAZyme stands for carbohydrate active enzymes. In fact, we didn't classify them themselves. We asked Bernard Henry Sett, who is in charge of the CAZyme database, and his team to do it. So we're more accurate about we're more confident about the classification of these proteins. But again, we see the same thing. And here we have the Acutatum species, members of the Acutatum species complex, and you can see that they're overrepresented in these genomes compared to the others, and also the Gliosporoides and the Orbiculari species complex. In fact, this is represented just by Colletotrichum orbiculari, which infects Cucurbitaceae. So I suppose it could be classified as a sort of a transitional or intermediate host range, or perhaps a broad host range. But all of these have much larger numbers of the carbohydrate active enzymes. We did the same thing with the proteases, this time using the MIROPS database, which is a database of proteases. And we see the same thing. So really, the members of these species complexes have a larger diversity of proteases and carbohydrate active enzymes than other species. And so the one question is, how did this situation evolve? And we form two possible hypotheses. Hypothesis one is that the lineage-specific expansions occur independently in the two lineages, once in the Acutatum lineage and once in the Gliosporides lineage. And if that's the case, then this is an example of convergent evolution. Hypothesis two is that these are ancient expansions in a common ancestor, followed by gene loss in all of the other species. So how did we test these hypotheses? Well, we constructed some phylogenetic analyses of 6 CAZyme gene families and 4 protease gene families. And I'm not going to show you all of those, I'll just show you 2. This is a phylogeny of the acetylesтерase gene family. These enzymes are responsible for hydrolysis of xylans, and you can find highly acetylated xylans in woody plants, okay? And I want to point out just a couple of things. I don't know if you can see this triangle, hopefully you can, but it's highlighting this clade, starting with this node, this whole clade. And this whole clade, the phylogeny of this clade basically reflects the evolutionary relationships among the species. And we don't observe in this clade any gene duplications or gene losses. Okay, so that's sort of a reference. We can switch to this example here, highlighted again with a blue triangle. And we only find this members of this clade in Acutatum, and let's see, there's members of the Gliosporides complex in here. But all of the other Colletotrichum and all of the other species included in the phylogeny are missing from this clade. So apparently this gene was lost in all of these other species. And in fact, if you go around the phylogeny, you find many examples of this. Here, in fact, just about all of these clades, we find gene losses in all of the species except for the Acutatum and Gliosporides species complexes. So what does this mean for us? It appears that these events were formed by, or the gene content in these species is due to an ancient gene expansion in a common ancestor of Colletotrichum, or perhaps before then, followed by gene losses in multiple lineages. Okay, so this is consistent with our hypothesis 2. And again, we find the same thing with the protease families. This is the M43B metalloendopeptidase family. And again, if you look at the different clades, you will see that we have expansion, or most of the clades contain members of the Acutatum species complex, Gliosporides species complex, but many of these clades have been lost in the other members of Colletotrichum and all of the other non-Colletotrichum species that we have. So again, this also suggests that there was an ancient expansion followed by gene loss. So we can say that the diversity of the CAZymes and proteases that we find in Acutatum and Gliosporides seems to be associated with a broad host range, and that there were ancient expansions followed by gene losses in some species. But the Acutatum and Gliosporides species complexes have maintained the diversity in these families. And this is not a case of convergent evolution. It also suggests that if the common ancestor of Colletotrichum or some other ancient Sordaria mycete had a larger diversity of CAZymes and proteases, perhaps it was also a broad host range pathogen, and that some of the narrow host range pathogens became host specific, and that coincided with the loss of this diversity in the protease and carbohydrate degrading enzymes. So now, what are we doing now? We are starting to focus on just the Acutatum species complex that you can see in this phylogeny. So this species complex contains more than 30 species, and those are divided into at least 5 main clades that you can see labeled on this slide. What's interesting is that while most of these are dicot pathogens, there are some monocot pathogens in here. There are mostly broad range pathogens, but it also includes a few intermediate and narrow host range species. So we can do the same kind of study in Acutatum, but on a much finer evolutionary scale. And we're hoping that this kind of study will help us to understand the process of gene duplication and gene loss in the evolution of host range and host preference. So to arrive at that point, the first thing that we did, or that Ricardo did, was to sequence the genome of Colletotrichum orchidophyllum, which is the closest ancestor to the Acutatum species complex. We also, during this time, had been sequencing more Acutatum species complex species. And through a project with the Joint Genome Institute in the United States, we resequenced 60 Acutatum isolates. We also did a transcriptional profiling experiment to study adaptation of monocot and dicot pathogens to their different host substrates, but I won't be talking about that today. But we have some interesting results there, and that'll be coming up in an upcoming paper. Now, this is another heat map of interpro terms on the horizontal axis and species on the vertical axis, just like we saw before. And this time we include the orchidophyllid genome. And the orchidophyllum genome, as I said, we could be classified as probably a transitional species. It infects members of the Orchidaceae, but that's all. And what you can see is that there these blue colors here indicate gene losses in certain gene families, and those are CAZyme families. But what's interesting is that we don't find gene losses in the proteases. So here's another partial phylogeny. It was too big to show the whole thing, but here's the orchidophyllum genome, this Colletotrichum orchidophyllum in this clade. But essentially what we could conclude from the phylogenetic analyses that we've done, that including Colletotrichum orchidophyllum, is the same thing that we saw in Acutatum. We find lineage-specific gene losses in orchidophyllum. So that's consistent with what we saw before the Acutatum and Gliosporidae species complexes. Okay, so now I'm going to change to a different topic and talk about intraspecific genome variation in Colletotrichum graminicula. And the two main things that I'm going to present is some of our early work to characterize the pan-genome and also to identify structural variants. So at the time that we did these studies, we had a reference genome for strain M1001, and we had sequences for 7 other isolates that are listed here. And this slide just shows you the phenotypic diversity that we find in these isolates. Some are more virulent or more aggressive, I should say, than others. They have larger lesions on the leaves. And over here, you can see the different, the diversity in colony color and morphology and growth rate on oatmeal agar and potato dextrose agar. And this little map shows the distribution of the different isolates that we've been able to collect. So this is a phylogenetic tree on the left that shows the relationships among the different isolates. And in the middle, we have a bar graph that shows the differences in lesion size that we find between the different isolates. The green matrix, or the matrix over on the right here, represents the presence and absence of different genes. If there's a green line, that means that there is a gene that is present in this isolate, but it's absent in this isolate, for example. And the main thing that you can see here is that, as it says here, phenotypically, or based on lesion size, very similar isolates can have large genetic differences. And if we compare M1001, that's this one, to 113173, that's this one, these two don't have any statistically different differences in lesion size, but you can look at the pattern of gene presence in both of these, and you find that there's a lot of differences. There's a lot of genes that are present in M1001 that are missing in 1173. And we can find that phenotypically different isolates have very few genetic differences, like comparing 113173 to 63127, which is non-pathogenic. We find that, well, they're not identical, but the pattern of presence and absence is very similar. Now, for the core genome of the pan-genome and the core genome of Colletotrichum graminicula, what we did for this analysis is, well, basically, when we sequenced the genomes of these different isolates, we had the intention of performing de novo assemblies, but the assembly quality varied a lot between different isolates. So we treated the data like we would do with a resequencing experiment, and we aligned the reads to the reference genome, which is M1001. And then we looked at each gene, and if a gene had less than 50% coverage, then we consider that gene missing in that isolate, okay? That 50% value is sort of arbitrary. So we include here in this line in the table, those that have between 50 and 90% of the gene covered. So with this, we can identify which genes were lost in the different isolates. And for the reads that did not align to the reference genome, for this isolate, for example, we performed a de novo assembly of those. And then we looked for genes within the contigs that came out of that de novo assembly. And those we could consider to be dispensable genes because they're not at M1001, and we can determine whether or not they're in the different isolates. So first of all, we have the core genome of M1001, which is 12,006 genes. And if we subtract the 141 genes that are missing in at least one of these isolates, we arrive at 11,865, and that is our core genome. And in there is where we would expect to find basic housekeeping genes, okay? And then we compare all of the genes from the graminicula genome to the other Colletotrichum species, and we find 443 that are not in other Colletotrichum species. So these are the genes that are unique to Colletotrichum graminicula. In all of those de novo assemblies that we did, we found 152 that are not in M1001. Then the pan-genome is the sum of these two numbers, the number of genes in M1001 plus these new genes. Another way to well, first of all, let's look at the genes that are unique to M1001. There's 11 of them here. Okay, these are genes that are only found in M1001. And we have an annotation for them. And what you can see is that 6 of them have no known or predicted function. They're hypothetical proteins. 4 of them are expressed either in planta or in Aprosauria based on looking at the RNA-Seq experiment that was published in the genome paper. 7 have no expression in Aprosauria or in planta. But what's interesting is that all of them are small proteins. But what's also interesting is that none of them have a secretion signal. So we thought that by looking at these unique genes, we might be able to find candidate effectors. But that turns out not to be the case, because if they don't have a secretion signal, they're not likely to be an effector, because an effector should be secreted out of the cell so that it can interact with the plant. Okay, so getting back to this issue of the pan-genome, this is a couple of graphs that represent, which is another way to visualize the pan-genome as a function of the number of genomes that you're looking at. Okay, so as we increase, we can look at the blue line, which are the dispensable genes, the genes that are not present in every isolate. And as we add more genomes to the analysis, we can see that we discover more dispensable genes. And we can see that the curve, as we approach 8 genomes, is starting to become level, but it's still not level. So as we add more genomes, we expect this to continue to increase. And the core genome are the genes found in all of the isolates, and that has the same pattern. So it's continuing to decrease as we include more genomes in the species, in the analysis. So now we have genome sequences for 20 Graminicula isolates, and we're repeating this analysis now, and it'll be interesting to see if this line levels off, or if it continues to increase. If it levels off, it would imply that we have what is called a closed pan-genome. And if it does level off, that means we've sequenced enough isolates to identify all of the genes that are present in Colletotrichum graminicula isolates. In contrast, if it continues to increase, then we have what is referred to as an open pan-genome. And that means that every time basically every isolate of Colletotrichum graminicula possibly has its own unique set of genes in it. On the right, this is simply a measure of the, basically it's another representation of these data, but it represents the number of gene clusters that are newly discovered as we increase the number of genomes. And it shows a similar pattern. It's still decreasing, but at some point it may level off. All of this means that there are more dispensable genes yet to be discovered. Now, regarding the structural variants that we could find in these genomes. So for this analysis, we used the mapping assemblies, the reads that were mapped to the M1001 genome. And you can identify structural variants with these data by looking at how the 2 reads, the genomes were sequenced with paired reads, how the 2 reads align to the reference genome. If they're oriented, basically pointing at each other like my fingers are, and they're at approximately the right distance according to the library size that was sequenced, then that indicates there is no structural variance. But if they're too far apart, or if they're too close together, that could indicate insertion or a deletion in that region. And if they're not oriented exactly like this, if they're oriented kind of like this, that could indicate an inversion. And if they're really far apart, maybe an interchromosomal translocation, etc. So this is the kind of information that could be gleaned from looking at the mapping of the reads to the reference genome. So on this plot, we show the 13 chromosomes that we found in the reference genome. This histogram with the green peaks shows the distribution of the genes in the genome. And the red peaks show the distribution of the repetitive elements. And the main thing that I want to point out is that the repetitive elements are sort of all over the place. They're not clustered into one region in each chromosome or something like that. So we find them all scattered all throughout the genome. Now I'll just zoom in on chromosome 1 so we can look at it in more detail. These histograms down here represent the distribution of structural variants. And the two things that I want to point out is that, first of all, there are a lot of structural variants. Structural variants seem to be very common based on this analysis, and that they're not compartmentalized as they are in some other fungi. Okay, so they seem to be scattered all across the chromosome as well. So this table and this figure basically show that there's no relationship between sequence similarity at the genome level and the amount of rearrangements. So in this axis, you can see the number of translocations between each pair of sequences or each sequence aligned to the reference. And on this axis, the sequence identity. Okay, and there doesn't seem to be any pattern to the intrachromosomal or interchromosomal rearrangements. Even the closely related species seem to have high numbers of rearrangements. Another interesting thing we did is to look at the distribution of the putative effectors in Colletotrichum. There are 5 effectors that have been identified and characterized in Colletotrichum graminicula. And what you find in some fungi or in some plant pathogens, like in Phytophthora, for example, they describe what is called a 2-speed genome, in which the effectors are in compartments of the genome that have unusually low gene density. So you can measure gene density across the genome by looking at the intergenic distance length. Low density regions will have large intergenic regions. And that's what we did here. We made a histogram of the length between each pair of genes. And then I decorated on this figure the locations of the 5 known effectors in the genome. And what you can see is that most of the effectors are right up here in rather regions that have rather, I would say, normal intergenic distances. And even these 2, which have slightly larger intergenic distances, are still in this region that I wouldn't consider to be very low gene density, which would be these regions here. So there's no evidence that effectors are found in gene-sparse regions of the genome. And in fact, based on this analysis and the previous analysis, we don't really have any evidence for a 2-speed genome in Colletotrichum graminicula. So let me just summarize this last part that we were just seeing. Basically, as I said, that these phenotypically similar isolates can have, or basically there is no correlation between genetic distance and phenotype of the lesion size, phenotype. The unique genes to M1001, we can't identify those as candidate effector proteins, which is what we were hoping to do in that analysis. The effectors, and essentially we have no evidence of a 2-speed genome, and we have no evidence that the effector genes in Colletotrichum graminicula are found in gene-sparse parts of the genome. And I should mention that essentially all of these analyses that I just presented we're repeating now by using PacBio genome sequencing. And we're resequencing several of our Colletotrichum isolates using PacBio. We get much better assemblies, and we'll have a much better picture of the actual distribution of structural rearrangements and so forth with the new assemblies. So lastly, I want to present some of our work to study the population structure of Colletotrichum graminicula. We wanted to determine what is the population structure. Is there evidence for migration among populations? And is there evidence for recombination? For this, we used a technique called RAD-Seq. RAD-Seq stands for Restriction Site Associated DNA Markers. And this works by, if you take a genome, like this line here, you cut it with restriction enzymes, and then you can ligate adapters onto those fragments. There's a size selection step, ligate adapters onto it, and then those fragments can be sequenced with Illumina, either with single-end sequencing or paired-end sequencing, and then those reads can be aligned to a reference genome, or they can be assembled de novo, but we align them to a reference genome. And the reads tend to form stacks like this. Okay, in fact, there's a software for analyzing these data called stacks. And with these reads aligned to the reference genome, you can identify polymorphisms, single nucleotide polymorphisms. And why do we do all of this? Because this technique is much cheaper than shotgun sequencing of the whole genome. Okay, so in our case, I think we achieved about 10% coverage of the reference genome using this technique. But that's enough because we can identify enough SNPs. With those data, we identified thousands of SNPs, and that's more than enough information for the population studies that we wanted to do. So we sequenced 95 isolates using RAD-Seq. As I mentioned, we have 20 isolates sequenced using shotgun sequencing. We can actually combine both of these data sets together to identify polymorphisms in the genome. And this figure down here just shows the geographic distribution of the isolates that we included. Most of them are from Europe, North America, and South America. So we covered about, with the RAD-Seq reads, we covered about 10% of the genome, of the reference genome. And if we look at SNPs that are present in 90% of the isolates, or should I say that we have read information from more than 90% of the isolates, we have almost 10,000 SNP polymorphisms. But if we look at the ones in which we have read information for 100% of the samples or 100% of the isolates, then we have 6,510 polymorphisms. And these are the data that we used for what we're going to see on the next couple of slides. You can see in this figure, this line here is the reference genome. This is just an example of some of the SNPs that we find. You can see how the reads stack up. If you zoom in, you can find the restriction sites in the genome sequence for the restriction enzyme that was used. These arrows point to a little line that you can see going up here. That's a polymorphism. There's another one here. I can zoom into one of these examples and you can see the polymorphism right here. Here's the stack of reads. This isolate has a T in this position, whereas the reference genome has a C. Here's another example here. So that's just some information about how we obtained the data. So one of the first things we did is called the genetic clustering, in which we compare the, we try to fit the allele information to a mathematical model, which represents different numbers of clusters. And clusters represent, in this case, populations. And for each cluster, we can measure the fit of that mathematical model to the data with a value called the Bayesian information criterion. And what we tried to do is find the number of clusters or populations that minimizes the BIC. And that would be this value here. And that is a value of 3. So this experiment suggests that there are 3 main populations of Colletotrichum graminicula. Here are the results of a principal components analysis. And this, interestingly, suggests that there are three main populations in South America, North America, and Europe. And I just want to point out a couple of things. First of all, this is the European population. And these are all European isolates, except for a few here. Those are isolates that we obtained from Argentina. So why are those isolates clustering with Europe? They perhaps migrated from Europe to Argentina. How could that happen? Well, these isolates were collected from a region in Argentina that was used, maybe it still is being used, by maize breeding companies who use Argentina and South America as a nursery to advance their breeding material another generation in 1 year. So one possibility is that some of the plant material that they brought from Europe to establish their winter nurseries was infected with European Colletotrichum graminicula, and perhaps it escaped. So we're obtaining more in isolates from outside of this region in Argentina to include in another RAD-Seq experiment. So maybe we can shed some more light on the origin of the Argentinian isolates. The other thing that you might want to see is here we have a South American isolate that clusters with North America, and a North American isolate that clusters with South America. What happened there? Well, we don't know. We only have one isolate here and one isolate here. It could have been cross-contamination in the lab, mislabeling samples. In fact, in the publication we're preparing, we're going to remove those because we cannot exclude the possibility that those are from contamination. So, in fact, maybe I shouldn't present that in a public meeting, but since it's just us, I think it's okay. But actually, we may try to re-isolate those and resequence them to confirm whether or not this isolate is actually from North America. And we come up with a phylogenetic analysis based on these SNPs. And again, we see three main populations. Here's the European one, the North American one, and the Brazilian clade here. So all of this information supports three main populations that are associated with three different continents. We did an analysis with by constructing a minimum spanning network, which is shown here. And in this, it's important to note that some of these circles, the circles represent the isolates. The larger circles represent more than one isolates that are genetically identical. Those are called multi-locus genotypes. And what's important here is that, well, first of all, we do find the three main clusters here of South America, Europe, and North America. But the other main point here is that we found relatively few multi-locus genotypes. The largest multi-locus genotypes only had 4 isolates in them, and I think there were only 2 of those. The rest only had 2 or 3 isolates in them. So what that means is that there's very little clonality in these populations. There's a lot of genetic diversity in them. And I'm almost done here. This is a phylogenetic tree that's generated with a splits tree. It's more of a network. It's more of a network. Phylogenetic trees are normally bifurcating, but you can see that these have more than one, can have multiple branch points. And these are called reticulations, and these are consistent with populations that are undergoing recombination, okay? In fact, there's a test in the splits tree program called the Phi test, which states that the results of which show that there is significant evidence for recombination among these isolates. Also, another interesting analysis that we did is plot a linkage decay plot. And the green line shows the point at which linkage disequilibrium decays by half. And it seems like a relatively short distance to me. So there's rapid linkage decay. Also, what's interesting is that many physically close loci show no genetic linkage. So there seems to be a lot of recombination among the chromosomes in these populations. So just a summary of the things I've just shown you. We have evidence for recombination and little to no clonality, which suggests, well, again, recombination. And I put here sexual recombination with a question mark, because as we know, in fungi, sexual recombination is not the only form of recombination. There's also parasexuality. So we can't exclude that possibility, but there is plenty of evidence in these data for recombination. And that is significant because prior to this, the general thinking was that C. graminicula reproduced mainly by clonal reproduction. And that seems to not be the case. We have evidence for at least 3 main lineages that correspond to different continents, as well as a European origin for at least some Argentinian isolates. So on the question, I just want to show this picture, which was taken by Serenella Sukno some time ago. But she was able to cross 2 isolates of Colletotrichum graminicula and produce asci and ascospores, the sexual reproduction structures. So it is possible to have sexual reproduction in this species. So perhaps there is a lot of sexual reproduction going on in the populations that we didn't know about before. Okay, I'm going to finish there with this slide to just thank all of our collaborators, our students, and postdocs, our former postdoc Riccardo Baroncelli, who did a lot of this work, especially the work on the Acutatum species complex, as well as to thank our funding, our sources of funding for this. And for that, I'm going to stop sharing the screen. And I'm going to open the chat in case somebody wants to use that.
Okay, thank you, Michael. Thank you very much for a nice talk. And well, you know, anybody that wants to make a question can either just start speaking or write it in the chat. So questions? Okay, I have questions. I will start then. Well, you know, Michael, I think that Colletotrichum is a really, really amazing fungus, amazing genus. It's so,
thank you. Your fungus is wonderful too.
It's so diverse in the number of different species that it's been very hard to classify them. That this is why this complex species complex and also the different lifestyles and not within the pathogenic, also different necrotrophic, hemibiotropic. Well, do you think, this is a very, very, very general question and very philosophical. Do you think we know that there is so much diversity because Colletotrichum genus has been very well studied, or this is just a very special feature of this genus, and why do you think it is so diverse? Well, I don't know thoughts about it.
I mean, you also have to keep in mind that taxonomists play a big role in defining what is a genus. So, you know, some taxonomists may divide Colletotrichum someday into different genera. You know, so, you know, I mean, some of these things are kind of beyond my expertise. In fact, in Colletotrichum, there was a time when all the species were being, all the species that we have now were grouped into a smaller number of species. And then taxonomists started to go in and split up some of these based on phylogenetic evidence, mostly, I believe, went in and started to divide some of these large species up into what we have now, which are species complex. I think in Acutatum was originally just Colletotrichum acutatum. And then that was divided up into these different species. So what we call a genus is, it's based on a lot of different criteria. But in this case, we have a lot of morphologically similar and evolutionarily similar species. So it seems like, it's just, seems to be a large group. But maybe taxonomists will break up some of the, will group together some of the smaller genera someday, who knows?
Okay, thank you. Antonio Molina, I think he has his hand right.
Hi, good morning or good afternoon, and thanks for this very interesting talk, Mike. So my question is related with the CAZymes and this experiment that you mentioned that you are testing in different carbohydrate or carbon sources. So as we are, as you know, very interested in the cell wall and these enzymes. So what kind of carbon sources are you using? So yes, complex one, or yes, specific polymers, or
no? These were purified cell walls from. I think we used maize as a monocot, and I think it was sugar beet as a dicot. These were experiments done by Ronald de Vries, and the experiment was set up with 2 monocot pathogens and 2 dicot pathogens. And we grew both of those pathogens in vitro on these 2 substrates. And also, I think it was glucose that was also a 3rd substrate. And what we want to find out is there adaptation at the transcriptomic level for growing on the different substrates? So if we grow a dicot pathogen on a monocot substrate, do we find differences in gene expression as compared to when it's growing on a dicot substrate?
Okay, and these data are published or are unpublished?
It's unpublished, and hopefully will be published soon.
Okay, now because
we're working on the paper.
The type of extraction that you perform is just a general extraction of the cell wall, or just a different subtraction, or just to get the hemicellulose pectin? So
yeah, I'm not sure of that.
Okay.
I would have to ask Ronald de Vries about that, because they did the actual experiments.
Okay, thanks.
Fernando, you also has a question. Fernando García Arenal.
Yes, I have 2 questions from the first part mostly. So you are analyzing the evolution of the genome in relation with host range, and how was host range assessed? I mean, usually for plant pathogens and for animal pathogens as well, host range is really unknown, and we have data from experiments, but very few data from nature.
Yeah, so we have general knowledge about some species in both of those species complexes, and as well as some other species within Colletotrichum to give us an idea of what is the host range, but not all of the species. And that is one problem that we have. And we would like to do more experiments. We would actually like to take some of these species and put them on a wide variety of hosts to really get a better understanding of what is the host range. But what we do know about them is based on what we find in the literature. So so we know, for example, that Colletotrichum orbicularis infects cucurbits, members of the Cucurbitaceae, but there have never been any reports of it being a pathogen of anything outside of the Cucurbitaceae. So that's the kind of information that we use for a lot of these cases. Colletotrichum graminicula for example, we know is, or we believe is restricted to maize. There have been some experiments to try to infect sorghum with Colletotrichum graminicula. But you can only do that if the sorghum plant is very unhealthy. So it seems to be restricted to Colletotrichum. So there is some information in the literature, but there is still a lot of unknowns about that.
This was my pre-question. The question was that according to the losses of enzymes that you see, and that the diversity of the enzymes is proper of the broad host ranges, it seems that this group is evolving towards specialism, correct?
Well, in the Gliosporides and Acutatum species complex, apparently not, because they seem to have maintained a large diversity of these degradative enzymes. But in other lineages, yes, it seems that way.
Yeah,
it seems that way. And as I said, it's interesting to speculate that the common ancestor of all these fungi had a large diversity of these degradative enzymes. So therefore, it may have been a broad host range pathogen. And then in some lineages, it seems to be that they're evolving towards specialization.
mm-hmm.
In that case, well, being host specific, is that an adaptation? It could be an adaptation. Why would it be beneficial to infect only one host? It would seem to be a better adaptation to have more options, right? Yeah. Okay. That brings up a lot of questions, but
right. Thank you, Michael.
Yeah, Sandra. Sandra?
Hello. Hello, can you hear me?
Yes.
Hello, Michael. This is Sandra Diaz.
Hi, Sandra.
I have a question regarding the CAZyme gene content in the hemibiotrophic fungi. And as you know, or you may know, Colletotrichum tophieldi, which is an endophytic fungus that is beneficial in very large or a large host range, is also encoding for a large number of CAZymes, and my question is if you have thought about doing a comparison of those hemibiotrophic or pathogenic Colletotrichum and other endophytic or neutral Colletotrichum that could also encode for a large number. You included only pathogens and saprotroph, but not beneficial or endophytic fungi.
Yeah, that would be interesting. In fact, maybe in the case of Higginsianum, we see a large diversity of CAZymes in this fungus because even though it's endophytic in a wide array, but for the same reason, because it needs to be able to use a wider diversity of substrates as nutrient source. But yeah, it would be interesting to look more at to compare endophytes versus pathogens. One of the problems I see could be that some fungi are pathogens on one species and endophytes on another. And we find quite a few endophytic fungi in Colletotrichum, even graminicula. We've demonstrated in a paper from, well, a while ago now, maybe 15 years or more ago, that we can infect the roots of Colletotrichum graminicula, the roots of maize with graminicula, and it will grow as an endophyte, essentially, and it will grow and colonize the plants, and eventually it'll become a pathogen again. But for some time, it's an endophyte. So, yeah, I guess the problem there is how do you define what is an endophyte and what's a pathogen?
Yeah, that's true. and also So I had another question, and it's related to the different populations that you found in your RAD-Seq analysis. And even you think that maybe you have a cross-contamination, or could have a cross-contamination, or something like that. Do you think that is possible that those results are okay, so you didn't have a mistake or a cross-contamination or something like that? So it is possible that you find a South American isolate grouping or clustering with the North American and so on?
Right, well, that's possible. The problem is if we only have one isolate, the contamination hypothesis is also strongly valid, right? So if we wanted to say that there is migration between South America and or Europe and Brazil, for example, or Europe and North America, right? Then I would like to have more isolates, more individuals to support that.
So as in the case of, so as in the case that you have a group of South American isolates grouping with the European, so for that you give so credibility? Right.
We give that credibility because we have multiple isolates that show the same thing. So we are, I can be more confident in rejecting the hypothesis of contamination in that case.
Okay, thank you very much, Thon. And nice to see you again.
Yeah.
María Ángeles Ayllón.
Yes.
Hi,
Michael. Nice to see you. Great talk.
Yeah, thanks.
I was wondering about this Colletotrichum graminicola isolate that there are very different in phenotype, and that this phenotypic difference are not related with the genetic distance.
Right.
Do you think that can be the phenotype can be influenced by the presence of mycoviruses, as was described for other species of Colletotrichum?
Well, it's certainly possible, isn't it? We haven't looked at for microviruses in any of these isolates, but yeah,
I was wondering, since you have this large amount of transcriptomic data, maybe you can also find mycoviruses associated with this isolate, because it has been already so that some mycoviruses associated with another species of Colletotrichum produce hypovirulin.
Yeah, that's a really good idea. I didn't think about that. So we have several transcriptomic data sets now. They're all done using one isolate, but at least for that isolate, it's isolate M1001, which we use for all the lab work. But yeah, it would be interesting to look through that to see if we have any viral-like sequences.
Exactly. Okay, thank you.
I do have some more questions, Mike.
Yep.
You found in these 11 strain-specific genes of this reference genome that most of them do not have secretion signal, although they look, otherwise they would look as effectors because they are small proteins, they are expressed in planta, and also the fact that they are lineage specific also points to that function.
Right.
An alternative way of being secreted, or otherwise what is your hypothesis about these genes? Do you have any thoughts about it?
Yeah, well, it would be interesting to speculate that there is an alternative secretion pathway. I think there are a few fungal genes. I don't remember very well what species they were in, but were reported to be exported along with vesicles, I believe.
Yeah.
And they don't actually go through the ER, as far as I understand. So they would obviously wouldn't need a secretion signal peptide. So that's a possibility. Another possibility is that they were just not properly annotated.
Yeah.
So I described the process that we did to get to that point, and there is a lot of room for error when you are trying to annotate genes on very short contigs, which is mostly what we got out of the analysis of the de novo assemblies. So I think we'll have better luck with that when we get our assemblies of the other genomes, the other isolates that we're sequencing with PacBio. And I think we'll be able to confirm some of that. And I should point out, we haven't actually published any of that information about the pan-genome or the structural rearrangements, because I thought we really need better assemblies in order to support any of our conclusions on that. So that's still a work in progress.
And have you found families of effectors as it has been found in other fungi that you have defined families that look that there has been an expansion, or are they very different ones from the others?
Well, some of them, most of the ones that we're working on actually are members of conserved gene families. In fact, one of the effectors that we have worked with that we have already published is a serine protease, which is a large family of genes, but we haven't looked at any of the other serine proteases to see if those might be effectors too. But yeah, it's still another open question.
Yeah.